And my definition is that it's this joint design of your hardware and your software for imaging systems. So you have some like hardware toolbox where you have maybe optics, or maybe it's electron hardware, or maybe it's MRI. But in general, like you have some hardware design. This is typically the domain of optical engineering, which is a bit of a black magic. And then you take some data and you do some post-processing on the data. And so that's the computational or software side. And in computational imaging, very often the data you capture, which would be Y here, is very indirectly related to what you're trying to solve for, which might be X in this case. And so the solve for X means you're taking images or an image that looks nothing like the final result. But it has the information of what you're trying to solve for. And so you can then solve an inverse problem to get back at the image that you were looking for. So the newest trend in computational imaging is around data-driven design. So bringing in machine learning to figure out how to optimize these systems. And I'll talk a little bit about this, not just optimizing how you do that reconstruction algorithm, but optimizing your hardware, which is something that really hasn't been very possible until recently, except for simple systems because of computational requirements. So let me start with a canonical computational imaging problem that's related to one that from our lab that I'll talk about. This is a lensless imaging problem. So you take a camera and you remove the lens. It's just a sensor. You point the sensor at the world and take a picture. And of course, it looks like garbage. The lens wasn't there to form the image. But the light that hit the sensor is the same light that would have hit the sensor if the lens was there. And so the lens's only job is to bend the light rays in a particular way. So if you can do this sort of ray bending computationally, could you take this image and reconstruct your scene from it because it's the same light? The answer is practically not really. This diffuser cam that we work on is basically showing that you can't just use only a sensor, but if you put something simple in front of the sensor, so this is just a bumpy piece of plastic, you can literally use the stickers that you put on your windows so your neighbors cannot see in, and just stick that on top of the sensor's cover glass and take a picture that also looks like garbage. But now, because we've modulated the light, you can think of it as modulating the angles of the light rays, then we have a structured garbage here that we can then reconstruct the image from. So we are messing with the light rays as they come in, but not in the same way as a lens. And this gets you a camera that's really cheap and compact because you don't need that space between the lens and the sensor. So if you think about comparing this to a regular camera is often characterized by its point spread function. This is essentially the impulse function of the system. So you put a point out in your world, in the scene, and what does the sensor measure for a point in the scene? So ideally, the sensor would just measure a delta function or a point. If we consider diffraction limited imaging systems, you'll see this airy pattern of the intensity. When we go to the diffuser cam, what happens is our point spread function, so we put a point in the world and there's nothing to focus it. And so it spreads out all over the sensor and then it goes through this diffuser, which is a pretty smooth piece of plastic. And you get this random focusing effect. It looks like the bottom of a swimming pool on a hot day, exactly the same idea. So 2 undergrads in my lab built this really cool tutorial so you can try building this at home if you like on our open source website, and you just need a Raspberry Pi sensor. These are like the cheapest sensors you can get, and some double-sided tape as your diffuser. It's not a good camera, but it's fun. And several undergrad classes have adopted this for labs and projects, and they've sent us some of their pictures. It's really fun to see what people get out of it. Okay, so our diffusers are a little bit more fancy and our sensors are definitely nicer than the Raspberry Pi. But if I have a point in the world, it gives me this caustic pattern impulse response function or point spread function. If I move the point in the world, then I get the same caustic pattern and it merely shifts. So we have a shift invariant system. If I turn on 2 points in the world, I get the linear sum of the intensities I would measure for each. So this system is linear in intensity, meaning I can write it in matrix form. So I can write it as a linear equation. So my measurement y is just the forward model, or this A matrix, times the scene in the world. So we raster scan the scene into a vector. And traditional cameras have spent centuries trying to make these A matrices, or the forward models, as close to the identity as possible, so that what you measure is what you're trying to take a picture of. When we talk about computational cameras, we kind of throw that idea out the window, and we say that as long as this A matrix is known and invertible, then this measurement y need not look anything like the scene that I'm trying to reconstruct. And indeed, for our diffuser cam, this is what our measurement looks like, and this is what the reconstructed scene might look like. So there's a problem here. I said we need to know this forward model A, and that's not so simple. So if you just put Scotch tape, or even the off-the-shelf diffusers that we use, we don't know the forward model. Furthermore, it's humongous. So if you have a 1 megapixel sensor, which is pretty conservative, then you have 1 million pixels in X and 1 million pixels in Y, meaning that the A matrix is 1 million squared. So you could brute force go out and measure that A matrix, basically put a point source in your scene here and measure the response. That will give you the first column of the A matrix, move it, get the second column, and basically measure column by column the entire A matrix. But then you're taking 1 million calibration measurements, and you're creating this million by million squared A matrix that you then need to invert, which is computationally a huge mess. Not to mention that being able to move a point source in the scene requires a very precisely calibrated motion stage, which can be very expensive. And we want you to be able to build this at home on the weekend. And so that's sort of not, that's not what we want. We could model this A matrix. So we could go and measure the surface shape of our diffuser and use that to model the A matrix. And that works very well. But again, not everyone has an AFM or an interferometer to measure the surface shape of their diffusers at home. And so that's also not practical. I'll talk later about this machine learning idea, but in practice, what we do is a combination of all three that allows us to get away with a single calibration measurement. And really, the heart of why we can do that is this shift invariant property, that as this point source in the scene shifts laterally, I'll just play that again, as the point source shifts laterally, so does the caustic pattern. So you have the same system response except shifted for shifted points in the scene. So the system is fully shift invariant. All the signal processing folks will know that that means this A matrix is convolutional. So every column of the A matrix is the same as the one before it. It's just shifted by an appropriate amount. So I will mention that these caustic patterns actually fly off the edge of the sensor at some point, and there's a cropping factor that needs to be accounted for. I'm just going to ignore that, but we do take it into account in our algorithm. But other than that, this becomes a deconvolution problem. And so we take our raw sensor data and run a deconvolution algorithm to get our recovered scene. And the calibration measurement is just this one point spread function measurement where we turn on our iPhone flashlight somewhere in the scene and take an image at the sensor. The way we're solving it in this case is with ADMM, and we're using a little bit of total variation regularization just to do some noise cleanup. I'll talk more about this later. This all runs in real time on my really old laptop. If we use Halide, Halide is a neat programming language invented by one of my colleagues that helps you use your GPU hardware really efficiently without being a fancy GPU programmer. Here's some more pictures and a video. And I like to show this video because you can see clear artifacts here. This is not competing with the quality of your iPhone yet. I think a lot of work could be done to improve the model mismatch problems that are causing these artifacts that could give you much higher image quality. But that's not really the direction we wanted to go because I just don't see this replacing your iPhone camera anytime soon, since iPhone cameras are already extremely compact and very cheap, and they already use plastic molded lenses. So what I want to talk a little bit about is the algorithm. So once you've got this captured measurement, how do we solve this inverse problem? And there's been a lot of work in computational imaging lately around what's the best way to solve this inverse problem by tuning your optimizer. So you use some sort of iterative optimization algorithm, and people have played around quite a bit with, priors or regularizers on the data, and this can make a huge difference in the quality of the result. So here's our inverse problem setup. We have our measurement y, and we're trying to minimize the difference between that measurement and the expected measurement of our current estimate of the scene passed through our forward model A, and it's convolutional. We have a constraint here that enforces positive light because there's no such thing as negative intensities. So that's just an extra constraint. And then this last term is our regularizer. And we can enforce some sort of sparsity. In the case that I'm showing you, we mostly use total variation only because we played around with lots of regularizers and this one seemed to work the best, but not necessarily always going to be the best. So this is what I would call the physics-based image reconstruction. We use an iterative optimization algorithm to solve for X, given measurements of Y, for a known forward model A. So physics-based in the sense that this forward model A captures the physics of the system. So we put in our measurement and get out our final result. The other way you could solve this is by throwing it at a deep neural network and giving it a whole bunch of input-output pairs and hoping that the network can learn the physics itself. So if you have enough training data and a deep enough network or a good enough network architecture, then you could learn these input-output relationships and be able to spit out the final result from your measurement using a deep neural network. So we've thought a lot about which way to go, like which one of these is better. And deep learning is popular right now, but it doesn't always give you better results. And it comes at huge costs sometimes because you often need very large training data sets, whereas this physics-based reconstruction doesn't need any training because you're inserting this prior knowledge about the physics of the imaging system. So if we think about, we were using ADMM, but sort of FISTA, gradient descent, all of these iterative optimizers to solve inverse problems. They're pretty interpretable and robust compared to deep learning, but they can be slow. And then model mismatching is definitely a big problem for us, that we take this calibration data and maybe, the camera heats up and something warps a little bit. It's not quite right. So machine learning is great at dealing with that, but it's not very interpretable, etc. Deep learning also has a massive improvement in the reconstruction speed once you've trained the network. So you put all the work up front in the calibration phase, and then once you've got the network trained, it's very fast. So what we really wanted was the best of both worlds. And so we started thinking about this physics-based learning, and a lot of these ideas existed in other areas. And so we're just bringing them into this computational imaging framework. But the idea is that you can use the physics that you know to help design the architecture of the neural network and then learn the stuff that you don't know. So you can get a really efficient neural network, and you can use that knowledge that you already had, which I find much more academically satisfying. So the way we did it is the unrolled algorithm. So we take our iterative optimizer algorithm and we unroll it so that each layer of the neural network is one iteration of this iterative algorithm. And then we can start learning all of the parameters of the algorithm and more. So here's an example. This is a ground truth image. If I do this physics-based optimizer, the result is has lots of artifacts and it's quite slow to compute. If I do pure deep learning, then I can calculate the image fast, but it's also got lots of different artifacts. And ours is the hybrid method where we're using this physics-based learning, and we can get reasonably fast reconstructions. This is speed up of about 20x with the least artifacts. And so this is a nice example of how physics and machine learning can work together. All right, so I want to go on to some of the things that we've done with this diffuser cam system, because I mentioned that it's probably not going to be a good photography camera. We just don't have the quality that you would expect from, a commercial photograph. And we decided that that was okay, and that what we wanted to do was use these cameras to do things that regular cameras cannot do. And 3D is a good example because a regular camera has a focus plane, and if things are in focus, they are sharp. And if things are out of focus, they're blurred. And once they're blurred, that is lost information, and you can't deconvolve it back out again. So you lose high spatial frequencies with defocus in traditional 3D image, in traditional cameras. And so 3D becomes sort of very difficult without having that information. When you think about a lensless camera like ours, there's no focus plane. There's just a different response from different depths. And those responses at different depths can actually still contain lots of high spatial frequency information. So here's an example. This is my diffuser cam, so diffuser and sensor. And I'm just moving a point in the world to different axial positions. So what you see happening on the sensor is that it's basically the same caustic pattern still, but now instead of shifting laterally, when I shift that point axially, it's scaling. So our point spread function is different for every axial position. That's good. That means we can distinguish what depth something came from. And it's predictable. So we don't need to take extra calibration measurements. We could just scale the one that we have at a particular distance. Okay, so we wanted to solve this 3D problem, and we're still going to use a single measured image. So say it has a 1 megapixel camera. So your measured image is 1 million pixels, but now I would like to solve for a 3D scene. So I'm going to solve for 100 different depth planes at full resolution, so 1 megapixel at each depth plane. So this makes this A matrix 1 million by 100 million in size. So that's brute force calibration and computation. This would all be insane if we didn't have these nice shift invariant models that I talked about before. But we still have a major problem because this thing is still underdetermined, in this case by a factor of 100. And the way we're going to go after that is with compressed sensing. I like to start talking about compressed sensing, thinking about typical images we take might be very compressible, and you might be able to compress them. So you might be able to take an image and fully represent everything that's in that image with 100 times less data by using some compression algorithm. And so this is usually done in post-processing. Compressed sensing is all about, can we just not take the data in the first place? And what's difficult about that is figuring out which data you need to capture to capture all of the information about the scene with that like less amount of data. So there's a lot of requirements for this. I'm just going to show you that this system happens to be amenable to compressed sensing. So how many people have heard of compressed sensing? Maybe you can like raise your hand in the participants window. Lots of people, I think. Yeah, so basically compressed sensing is this idea that you can solve an underdetermined problem if you insert a sparsity prior. So you're inserting prior information that the scene is sparse in some basis set. And just to show you that this system works well for that, we can take our raw image and simply delete 80% of the pixels. So randomly choose 80% of the pixels, throw them out, and do the reconstruction, and it still works pretty well. If I delete 90%, even 98% of the pixels, and I can still kind of see what's going on. This wouldn't happen with a regular camera. And the reason that this works is because a point in the scene maps to a lot of pixels on the sensor. So when I start deleting pixels on the sensor, I'm not deleting information about all of the information about any points in the scene because they're going to other pixels as well. So there's no reason why you would take a 2D image and just throw away pixels. What we want to really use this for is to take the full set of pixels in the 2D captured image and use it to reconstruct the third dimension. And so this is an example there. We capture this single 2D image of this little leaf, and this is the 3D reconstruction. I'm just spinning the reconstruction so you can see that we get good depth information here. So I say 128 more voxels for free because we're reconstructing 128 depth planes here at almost the full resolution. Okay, so what's really exciting about this for microscopy is to think about this trade-off between space bandwidth product and speed. So space bandwidth product is basically the number of voxels I can resolve within a volume. So if I want to look at a bigger volume with higher resolution, I'm going to need a very large space bandwidth product. And if you use scanning microscopy methods like confocal, they're just point scanning, raster scanning a single point through the 3D volume. If you want to look at more voxels, you need to slow down. You're going to take longer to capture them because you're just capturing one voxel at a time. Light field microscopy was exciting for a while because it could do single shot 3D imaging, but it did so at drastically reduced resolution. So their space bandwidth product was very low. And the reason you use a microscope is to get higher resolution. So it seemed like a lot of situations where it wasn't worth it to give that up. If we talk about this compressed sensing diffuser cam, then we have the possibility of getting both high speed and large space bandwidth product if the sample is extremely sparse. So basically this range here is about like how sparse is your sample. So 3D fluorescence microscopy is a great like candidate for using compressed sensing because you have to like physically tag these different molecules. And so it tends to be pretty sparse in 3D in the mathematical sense. So one of our applications that we're going after is trying to take like a big heavy microscope and make it compact and head mountable on mice so we can track in 3D their neural activity at millisecond speeds while they're running around and performing behaviors. We work with a neuroscience lab, Hillel Adesnik's lab, that studies, that decides sort of like which neurons they care about and what they want the mouse to be doing while they're watching its neurons fire. But this is a great candidate for that. This is a much bigger microscope that we use to show some proof of principle. This is a Zebrafish Brain. And what you see here is a reconstruction, a 3D reconstruction. So each point here represents the 3D position of an individual neuron that we detected. And then the colors over time tell you its activity during that time period. So this is basically dynamic 3D imaging. You could call it 4D if you think the fourth dimension is time. But this is really exciting because we're just taking a 2D video and reconstructing a 3D video. Here's the flat version of our microscope. This took a lot of engineering to get working. It's very hard to get color filters working when everything is so compact because you're coming in at high angles and the color filters don't work well at high angles. So this is neural activity in a zebrafish. These are individual neurons firing, you can see over here. And then finally, our last engineered design that I think is actually possibly the most practical is what we call Miniscope 3D. So Miniscope is an open source microscope hardware. You buy a little kit and you make this tiny microscope smaller than a quarter. It's got a grin objective lens that can be implanted in a mouse's brain. And it weighs only, well, the original one weighs a few grams. And the mouse can run around and go through a maze while you're watching particular neurons firing. So what we did is we hacked it to turn it into a 3D miniscope. And in the process of hacking it, we added this diffuser or phase mask right here it's at the pupil plane of this grin objective. And then we took out the tube lens. This was more of a traditional microscope design. We took out the tube lens, moved the sensor closer, so it's in this diffuser cam configuration. And in the process, we made the whole thing lighter weight and smaller. So this thing is only 17 millimeters and 2.5 grams now. So we get these like benefits of size and weight, and we get 3D information. And what is lost is about a factor of 2 in resolution, only because these are terrible objective lenses that have a lot of aberrations. And our computational reconstructions actually correct a lot of those. So here's the phase mask that we use. I'll talk a little more about it later. It's no longer just an off-the-shelf piece of Scotch tape. We're actually designing these things, and we can get down to a couple microns resolution across a pretty big 3D volume, which was the goal. And a couple microns resolution means that we can see individual neurons firing. So here is the raw data from a little tardigrade or water bear, and then the 3D reconstruction. You can see it's like a little caterpillar. You can see its legs here while it's crawling, and these are just 2 different perspectives on the 3D reconstructed video that we get. It's all reconstructed from this raw data. Okay, so now I want to talk a little bit about the diffuser. I used to give this talk and everyone would ask, well, what's the best diffuser to use? Can you design a better one? And yes, the answer is yes. So these smooth diffusers are what we started with. These are like the holographic diffusers from Edmund Optics. And they have nice like random focusing, which is good for the compressed sensing. So for compressed sensing, you want the light to spread out to lots of pixels. But for noise performance, you want the light to be sort of like concentrated in smaller areas. So this like hazy stuff in between the focused areas is bad. This is just like background. This is just making our SNR worse for this system. So a lens has the best SNR performance because all of the light from the point piles onto a single point here. But the lens only is the best at a single depth plane. If you're trying to do 3D, then a lens is actually the worst because you're spreading the light out and just smearing it, which is very bad for trying to deconvolve. So you could say, what if I use a micro lens array? Then I get good focusing. I get very black spots in between these dots. And I still get the light spread out, so I can still do these compressed sensing tricks. The bad thing about micro lens array is that because it's periodic, if I shift my point source by the appropriate amount, then I get a point spread function that's almost the same as the one. These two point sources have almost the same point spread function. So there's this ambiguity in which these are very hard to distinguish from each other because the thing is regular periodic thing. So sort of what we came out with heuristically is to say like, okay, let's use micro lenses, but we will randomly space them. And furthermore, we're trying to do 3D. So we're also going to give them different ranges of focal lengths. So different ones are focused at different points in the 3D volume so that we always have some sharp spots, meaning that we always get some high frequency information. So then, of course, the next step is to think about optimizing that more theoretically. So rather than just heuristically figuring out what's a better diffuser pattern, let's actually try to figure out what's the best diffuser pattern by doing an optimization. And this is sort of like a new trend in computational imaging. You can see all these papers are relatively new, but these are all papers around like what we call end-to-end system design. And this is about putting everything into a big optimization where you co-optimize both the parameters of the inverse solver, so like the regularization parameters, etc., and the hardware parameters. In our case, what we're trying to solve for is what is the best diffuser? What is the best, like, say I'm going to have some phase plate that's a piece of plastic that has a certain refractive index. What is the best surface shape for that phase plate that's going to give me the best reconstructions in conjunction with optimization of all those regularization parameters, etc., in our inverse problem. So this is a huge computational feat, particularly for 3D problems where you're trying to store a lot of the information. You just run out of memory. And Michael Kellman in my group and Eric Markley have been working on this, making this sort of practical kind of optimization to do. And so this is the diffuser optimization. So you're watching it optimize this diffuser shape. And what you see is as it gets towards the end, this is our like merit function, then we sort of like settle on having, I think there's 8 different micro lenses here kind of spread out in a circle. So it starts out with a lot more and it starts like killing some of them. And this is all optimized for 3D reconstruction of some point sources in a 3D space. You will get a different optimization if you feed it a different class of objects that you're trying to image, or a different range of volumes that you're trying to get good images in. So this is all designed to get single neuron resolution across the biggest volume possible. So I'm really excited about this. This is something that one of my newer PhD students, Eric, is working on for all kinds of different problems. And you'll see more of this later, but I just wanted to give a sense of it. Okay, so I'm half an hour in and I want to get into a few like offshoot projects that came out of this that I think are really neat and thought-provoking. So I've told you that I can take this 2D measurement and reconstruct a 2D image, or I can take this 2D measurement and reconstruct a 3D image by exploiting compressed sensing. So then we started thinking about what other 3D quantities could we reconstruct from this 2D measurement using compressed sensing. So basically, compressed sensing means we can potentially get an extra dimension reconstructed. And so the dimension that my student Nick went after, he's now faculty at UCSD, was time. So this was a really neat idea. So what he says, like, almost all our sensors are rolling shutter. So if stuff is happening faster than the rolling shutter can fly down the sensor. So rolling shutter means like you only capture one line of the sensor at a time, and it sweeps through the picture really quickly. And then your image is just like the sum of all of these line captures. So if stuff is happening faster than the rolling shutter can fly down the sensor, then you might miss some of it. So in this case, there was 3 things flashing, but we only captured one of them because it happened to line up with where the rolling shutter was at that time. This is an irregular lensed imaging system. In the diffuser cam, stuff is happening while the sensor is rolling its line down the frame. And there's always something about it captured because every point in the scene maps to the entire sensor. So no matter where in the scene something is happening, you get information about it on the sensor because it covers the whole sensor. So Patrick, who was an undergrad for the summer, worked with Nick on trying to solve this as basically a 3D inverse problem where you solve for 2D plus time instead of 3 dimensions. And what they could do then is take this single 2D image that was captured while something was happening dynamically during the rolling shutter's single frame rate, and they can take a single frame image and reconstruct 150 frames of video. This is an important scientific application of Nerf darts hitting apples sitting on textbooks. And this is really just a proof of principle. But I think this is really cool that they can reconstruct a video from a single still frame, or alternatively reconstruct a high-speed video that's allegedly 150 times faster frame rate than the camera's native frame rate with just a regular rolling shutter sensor. Okay, so then this was kind of cool. So let's think about other 3D things we can solve for. And Christina in my group went after hyperspectral. So instead of XYZ or XY time, she's going after XY lambda. And she had to change the architecture of the system a little bit. So it's still a diffuser in front of a sensor, but now we've placed this spectral filter array on top of the sensor. Spectral filter array here is from Viavi Solutions. They were really nice working with us. And they provided us with these like arrays of 8x8 different spectral filters that you just align with the camera pixels, and you take this coded measurement. And basically what we're after is to be able to solve for the 2D image and the 64 spectral channels at every pixel. And that's with compressed sensing, because otherwise you would have to either give up spatial resolution or spectral resolution. And this is the result for some colored checker cube, and you can see the spectral line profiles here. So this is exciting because it's so cheap. This thing is a $5 filter array that they can make with any spectral filters that you want. So we're working on like specific designs targeted at particular tasks so they can be much more efficient. One of the worst things about this is that it's sacrificing a lot of light because those spectral filters block a lot of the light. So you don't want to use extra spectral filters if you don't have to. So you can do this sort of data-driven design to make things optimal for a particular application, which is exactly what we're working on right now. All right, so that was sort of like all of these different projects centered around the diffuser cam. Now I want to take a few minutes to talk about a different project in my group that's much older, but then I'll put them together at the end. And this project was about coded illumination microscopy. So this diffuser cam stuff was all about fluorescent imaging, which is sort of like self-illuminated, or you flood illuminate things, and then these fluorophores light up, and then you image them. But now we're talking about non-labeled or like non-fluorescent samples, and we can start messing with the illumination to do interesting things. So we copied this setup originally from Cheng-Hui Yang's group at Caltech. They just put this LED array on top of their microscope. So it's just a traditional bright field microscope with the illumination unit replaced by this Adafruit LED array. We use much fancier LED arrays now. This was literally, Adafruit's a toy company, and you have a microcontroller to turn on whatever LEDs you want at different times. So this thing is just sitting above the sample, and as you turn on different LEDs, you're basically just illuminating the sample from different directions. So it's patterning of the illumination angles of the sample. Okay, so how is that useful? Well, actually, it turns out that this is a really neat hardware platform for computational microscopy. And by choosing different illumination patterns and using different reconstruction algorithms, you can get all kinds of different imaging modalities out of this single hardware system with no moving parts. I'm not going to get into it much. The reason we built this was to do this Fourier tachography idea, um, where, or gigapixel imaging is just another name for it, because it's all about going after really big field of view with also having high resolution. And normally you don't get both. You have to choose either zoom in and get high resolution across a small area, or zoom out and get bad resolution across a big area. So this gigapixel imaging or Fourier tachography, you start with a microscope that has a low numerical aperture objective, meaning that it has a big field of view, but poor resolution. So we're just not resolving the smallest features here. We do this disco pattern illumination that you saw in the previous slide. Take a video. So we take a video with all these different illuminations, and from this video, we can reconstruct. In this case, I think it's 7 times higher resolution in each dimension. So the reason we can do this is explained in Fourier space. So you need a little bit of Fourier optics. UX and UY are the 2D spatial frequencies of your sample. And because we used a low numerical aperture objective, it only captured spatial frequency information for the lowest spatial frequencies, hence the low resolution image that we captured. So every image we capture is going to be low resolution, in that it's only going to capture spatial frequencies from a small circle, this green circle here. But when I turn on LEDs that are far off axis, what I get is spatial frequency information from out here. So this looks like a dark field image because it's outside of the bandwidth of the illumination. And what you see is it lights up all of the vertical edges of the sample. If I light up an LED over here, I captured this set of spatial frequencies, which gives you all the horizontal edges. And all this stuff lighting up is sort of like sub-resolution information, but each individual image is still low resolution because it still comes from this small circle in the Fourier space. And so what you want to do is turn on each LED. Well, you can start by turning on one at a time. Each one captures the spatial frequency information from one circle in the Fourier space. And then I can just turn on lots of different LEDs, take a lot of different images from different parts of the Fourier space, and stitch them all together. This is called synthetic aperture imaging. The caveat is that we're only measuring intensity. And if you want to stitch things in Fourier space to get the higher bandwidth, which will give you higher resolution, you better know the phase as well as the amplitude. So we don't, and that's why these circles are all severely overlapping, because that enables you to do both a synthetic or aperture stitching in Fourier space and a phase retrieval problem simultaneously. So you can write this as a big optimization inverse problem. So this is like the model-based inverse problems I talked about before. And then you can solve for the larger Fourier space. We actually also solve for the pupil function, which contains all the aberrations in the system, because that gives us better images. And you can do this with these iterative optimization techniques. So the simplest one is gradient descent. If you've done phase retrieval, you've heard of Gerchberg-Saxton. It is gradient descent. So this is fast, but when you start to like reduce the amount of data that you capture, so we want to use the least data possible, you start to get weird artifacts. With the same data set, if you use second order optimization methods like Newton's method, you can get a much cleaner image, but it takes a lot longer to run. And this is only a tiny patch of an image, of a gigapixel sized image. We do about 200 by 200 pixels in a patch. So this is going to take forever to reconstruct the whole gigapixel image. And furthermore, we want to do videos at gigapixel. So here's some commentary on fast versus exact. But we don't have to choose. There's some sort of trade-offs here. And what we ended up using in the end was a quasi second order method, Gauss-Newton, which gives us pretty good quality results and is relatively fast. So everything I've shown you is using this algorithm to reconstruct from our data sets. Okay, here's some quantitative phase imaging. So when I say we do this phase retrieval, basically it means for free we get phase information. And so we can do quantitative phase imaging. And this is basically a map of the quantitative phase maps. For some HeLa cells, you can see this one is going to split, curl up in a ball, and then split into multiple cells. And this is the field of view you would have gotten with the final resolution that we reconstructed using our method, but now you get it across this much larger field of view. So you can look at a lot of cells at once. All right, so back to how machine learning can help. I want to get a little bit into how do we design those illumination patterns? So I mentioned you could just take one LED and turn it on and go around and do each LED individually. But if you want to look at live samples like these HeLa cells that I showed you, that is way too slow. You will get so much motion blur that you won't get any of the super resolution back. And so we started getting into multiplexing, meaning that we're going to turn on multiple LEDs at a time, and we're going to take fewer images. So our heuristic design was totally just based on knowing how the inverse problem works, knowing how the physics works. And we said, like, for these inner LEDs, so this inner ring of LEDs are all the LEDs whose illumination angles are within the numerical aperture of the system. So they pass through the system, you get a bright field image. All the ones outside here are called dark field because the illumination itself is at too high of an angle to pass through the system. So everything you get at the sensor is just scattered light purely. So in the bright field, it's really easy. We have this like DPC phase method. You just use these half circles. We know how that works. And then for the dark field, we just randomly chose 8 LEDs at a time. So we could take 8 times fewer images. And since there's 8 LEDs at a time, each image is 8 times brighter. So we got about a factor of 100 improvement in speed, which is what we needed to be able to do live samples. But then we started getting into this end-to-end design that I talked about before. And now we're just trying to apply it to everything we possibly can. And so we applied it to this choosing the LED patterns problem. And so we said we want to choose, I guess this is 8 different, we wanted to take 8 images. What are the best LED patterns to use for those 8 images? And this is what the machine learning told us to do. It's really interesting to look at this. So these two are the bright field patterns, which are not that fun. They're sort of like conceptually different than what we were doing heuristically. But then the dark field stuff is quite different than what we were doing heuristically. We're starting to try to understand it. I'd be happy to talk about that in questions if anyone's interested. But these learned designs performed a lot better. So if we took one LED at a time, we would have 89 measurements. So we consider this kind of like our ground truth. And then if we want to reduce to only 10 measurements, our heuristic patterns create a lot of artifacts, whereas the data-driven measurements give a much better result. So we can just get away with taking a lot less data. Okay, so then the last piece that I'm going to talk about here is about 3D. And so I've mentioned that we're illuminating from lots of different angles and trying to get super resolution. But of course, if your sample is thick, like a 3D sample, then illuminating from lots of different directions actually projects through the sample differently, like tomography. And so you can think about using different illumination angles to get 3D information. But actually, we can toss this all into a big optimization problem and solve for both super resolution and 3D information. It's totally analogous to training a neural network. There's a lot of papers on this. A lot of this stuff has been done long before us. But it's really exciting because this is like a neural network, nonlinear, non-convex optimization. We can't prove it will always converge. We can only heuristically look at when it works. But what's very exciting is that because we're modeling how light bounces around within the object, we are actually accounting for the multiple scattering inside the sample. So your sample has all these changes in refractive index, and we're accounting for how that scattering bounces around multiple times. So I want to be very clear how much scattering we're dealing with. Really weak scattering can be thought of more as aberrations. Very strong scattering, like totally isotropic scattering like fog, this is not something we can deal with. We're in this realm of like a little bit of multiple scattering. And if you think about tissue, this is in the realm of looking at things like small organisms. So we can't go like centimeters deep in tissue. We'll never be able to do that, too much computation. But for small organisms, we might have a chance. So I've been saying that we're solving for phase. So when you do 3D, phase becomes refractive index. So 3D phase imaging just means 3D refractive index imaging. And now I'm telling you that the 3D refractive index profile is very often called the scattering potential of the sample. So basically, if you know the 3D refractive index of the sample accurately, then you can predict exactly how light scatters through that sample, even if it's bouncing around and having lots of multiple scattering. So if you can measure 3D refractive index, you can computationally undo all scattering. And this is where you have to reconstruct the entire 3D refractive index to computationally undo the scattering. And so that's where like we're not going to get into very extremely high scattering cases because it's just too many degrees of freedom to solve for. So we might be solving for something like this. So this looks pretty speckly, means there's some multiple scattering going on, but it's not like total chaos. And here's some examples. Like a fibroblast cell, I would say, is not multiple scattering. If you take the Fourier transform of one of these illumination off-axis images, it has these two clear circles in it. Those are a total hallmark of single scattering. And so then when we use our single scattering model versus multiple scattering inverse problem, we get basically the same result because this thing wasn't multiple scattering. If I look at a C. elegans embryo, now these two circles are kind of blurred. There's information outside those two circles. That's all multiple scattering information. And now using the multiple scattering inverse problems gives us a much better result. We can pick up a lot more on the low spatial frequency content, which is what's really hard to capture in phase imaging. And if we go to even higher scattering, we kind of lose those two circles altogether. So this is a much more multiple scattering sample. And then you see an even bigger difference in the contrast and quality of the reconstructions with single and multiple scattering. And then I just want to show, this is a 15 gigavoxel reconstruction of the 3D refractive index of a C. elegans worm. And what I'm showing you is just a central slice along the center of this 3D reconstruction. And this was something we could never do before because the sample itself was scattering so much that when you tried to focus at the center of it, you just couldn't get a clear picture. But now after all of this computation to computationally undo the scattering, we can get some clear images of all of this stuff that's happening inside the sample. And so our next step is to go after more scattering samples. And embryos are great because embryos are just always growing and getting more scattering every day exponentially. So if you look at a fruit fly embryo at a certain stage, it looks almost opaque because it's so highly scattering. And in fact, even a confocal microscope can't get through the center of a fruit fly embryo at this stage because there's too much multiple scattering in the sample. So Chateau is going after this sample, and here's his 3D reconstruction. You could see it's still pretty hazy near the center, so we're not correcting everything, but he's still getting some structures in there. Our next step is to try the same thing with just a longer wavelength and some slightly fancier algorithms to see if we can get rid of that haze. But I think that it's really exciting to think about putting these projects together. So the diffuser cam stuff was all about 3D fluorescence imaging. And then this coded illumination is all about 3D refractive index imaging. But the 3D refractive index imaging tells you about structure, it tells you about scattering. And the fluorescence imaging tells you about function. So you can put these together and think about using this 3D refractive index profile, which is the scattering potential, to correct scattering errors in your fluorescence 3D imaging. And there's just a really quick example of something that Yi is working on to do exactly this, that she can reconstruct the 3D refractive index or scattering potential and use that to correct the positions of fluorescent beads to get them more accurate within her 3D fluorescence images. And so putting these modalities together can be really valuable. So that's all I'm going to talk about. We have a lot of stuff on our open source website if you want to try any of these methods. We've also have a small startup company that sells these LED arrays. These arrays are much more practical than the original Adafruit ones, and I'm part of this company. So if you want to try it, it becomes really easy to try. And I will thank my group for doing all this work and happy to take any questions.
Okay, so thanks a lot, Laura, for the great talk. It was really, really awesome. So we have a few questions. So just before I forgot to mention, so the talk has been recorded. It will be put on YouTube as well, so you can rewatch some highlights if you want. And so for the question I've asked, let me just unmute Jonathan. Where is he? He's on mute. So I've asked one of our local experts in computational imaging to select a few of the relevant questions because we actually have many. So Jonathan, you can go with the one you have selected.
Yeah. Hi Laura, thanks for this.
Hi, how are you?
Talk. And there are questions that you have already answered, a bit like the optimization of the diffuser. But one question which is quite interesting is the one on adversarial attacks. Like, do you think adversarial attacks are possible in your methods as well? And how reliable, how robust are the techniques?
I'm not sure I understood that question.
Like, adversarial attack is if you change a little bit the camera image to completely change the reconstruction. Like a small change in the camera image will change a lot the reconstructed object.
Yeah, we haven't done some very careful analysis of this. I think it's really exciting, like some of the like uncertainty quantification techniques that could be brought in here. We haven't done any of this. Adversarial attacks, it depends, right? So I always talk about like in phase imaging, because it's a nonlinear, non-convex problem, often the data you put in matters more than the algorithm you use as far as like whether it works or how well it works. And so I think that's where like, I suppose you could probably construct situations where you could have an adversarial attack, but I suspect that, you know, like an embryo with all of its complicated things going on, it would be quite difficult to do. I'm sure that there's multiple solutions and we're sometimes getting local minima that are giving us wrong solutions sometimes. But that's where like we haven't done a proper analysis because it's so difficult because it's a nonlinear system. But you should be thinking about this in terms of like, how many images do I need? What illumination angles do I need? Is this problem actually solvable? How much scattering can I solve for? And if you try to do too much, you're surely going to get the wrong answers. And there's going to be multiple answers that fit your data, possibly. But we don't have good analysis on that, only because everything is so nonlinear. So we do the linear analysis on it, which is not very satisfying, but it's a good start. But yeah, I don't have a good answer.
Thanks. And a question which comes a bit more from machine learning, I guess, it's you are talking about translation invariance, but how about rotation invariance? Could you add something like this to help when the system is underdetermined?
So rotating the sample?
In diffuser cam, if, for example, in a physical model, you add the fact that you have a rotation invariance. A bit like lens, for example.
So I don't know that we do have rotation invariance. We're thinking about 3D in terms of like just looking from different angles. I'm not sure if that would even be true.
I see. Okay, thank you. And another question, which is quite nice for imaging at EPFL, since like it's bringing different imaging modalities together. It's one asked by an astrophysicist. And in his case, the diffuser can be thought as a dark matter that sits in a galaxy between us and a more distant source, like another galaxy, for example. And since in this case, the universe is a unique experiment that cannot be recreated, then they have a lot of spectral information, for example. And do you think like the diffuser optimization techniques can be used also in this setting? And how well can it be to reconstruct the diffuser, which is the dark matter, from the captured images?
Yeah, if what you're after is, okay, so it might be pointless to try to optimize the diffuser in that case because you can't change the dark matter. Yeah. But if what you're after is to solve for the diffuser, you can certainly do that. You probably need some sort of diversity. So we've done this with like defocus diversity, or if you come from different angles, maybe that's something that could be possible in this situation. Or you can make priors, like if you know the distances of things approximately, and you know there's like empty space in certain parts, you could potentially do that. It's just setting up the problem differently. I don't know how solvable that particular problem would be.
Quite interesting, the directions.
Yeah, so the wavelength could be the diversity that you need to make it work.
Yeah, I see. And another question, like for the last part that you said with fluorescence and scattering potential, how do you use the scattering potential to correct the fluorescence image? And is there like a computational efficient way to do it?
I don't know if we have the most computationally efficient way, but it's pretty simple to think about. Like if you have like the 3D refractive index profile, and then say you have like a 2D fluorescent image down here, it's going to be warped by passing through this 3D refractive index profile. So like one really easy way is to just backward propagate the output light to the image plane that you care about, and then that's your final result. It should automatically undo all of the scattering effects. There's probably better ways to do this depending on, like, you know, if you have just a few sparse fluorophores versus a 2D image under a 3D refractive index chunk. We're just getting started with this, so I'm not sure if there's better ways.
That's interesting. Yeah, there are actually quite a lot of questions, but let me ask you another one. Especially for the application of machine learning to find optimal illumination patterns, are there other aspects of wave tachography where you see machine learning playing an important role?
I think that what I've seen is its primary benefit is making things faster. So putting all of the effort and time into the training means that you can have this neural network that can solve the result faster, which we haven't cared about too much because this is all like researchy stuff. But in the real world, people want to see the result of their image right away so they can decide what experiment to do next. And so getting them that result faster, I think is what it's been most useful for. It's also good at removing weird model mismatch artifacts, but it comes at a big cost having to do this like training data sets. So it really depends on your application.
I see. And maybe one last question, which is more like high level to conclude. So what do you think in computational imaging is the limiting factor today, or what excites you the most? Is it like developments of new ideas, understanding better the algorithms or the hardware?
I'm really excited about this end-to-end stuff. So being able to optimize everything, it just feels very satisfying academically to not be saying like, oh, we're designing this optimal inverse algorithm, but we just randomly chose which data we're going to put into it. Or like, you know, trying to think heuristically or using like expertise only. I think it's really exciting to be able to like let the computer figure out the best hardware or the best way to capture the data. And it might come out with things that seem crazy, that don't make sense, but work better. So I'm really excited about that. And the biggest challenge there is compute, is that we run out of memory. It takes forever to compute these things. Doing these optimizations, they're just so huge that that some of them become really unfeasible, particularly for the 3D problems. So we've been playing around with all different ways to try to improve that. Michael Kalman had some nice papers on using, if you can back propagate through the network, you need not store everything, which is really nice. That saved us a lot. It sort of like drastically improved the size of problems we could solve so that they're more practical. But we still run into these problems. And right now we're playing around with the solution of just throwing money at it by putting it on Amazon Web Services. But I'm not a big fan of that solution.